Hello dear students, welcome to EPG Pathshala. I am Dr. Veda Krishnan. I am a scientist in the Division of Biochemistry, Indian Agricultural Research Institute, that is IARI, New Delhi, India. I am going to talk about the overview of protein-mediated transport from the paper Protein Biochemistry and Enzymology. In cellular biology, the term membrane transport refers to the collection of mechanisms that regulate the passage of solutes such as ions and small molecules. These are facilitated by proteins and hence known as protein-mediated transport. Proteins, as you all know, one of the four major biomolecules in every cell, which carry out immense cellular functions like DNA synthesis, response to stimuli, mediate signaling, and also transport molecules in and out of the cellular machinery. A membrane transport protein, commonly known as a transporter, is a membrane protein involved in the movement of ions, small molecules, or macromolecules across a biological membrane. Transport proteins are normally integral transmembrane, that is, they exist permanently within and span the membrane across which they transport substances. In this module, let's discuss on various types of protein-mediated transport, or in a way we can say facilitated diffusion for micro and macromolecules in detail. Protein-mediated transport systems are of two types: system for the transport of micromolecules and system for the transport of macromolecules. Let's start in detail with mechanism of transport of micromolecules. The two major transporters in the system are channels and carriers. These are the types of facilitated diffusion which mediates the transport of molecules in and out of the cell. Let's see what are the major difference between these two. Channels can exist either in closed or open state based on the stimuli or conformational switch, while carriers cannot be opened simultaneously like channels. For carriers, either inner or outer gate will only be open at a time. Another major difference between them are channels have no binding site while carriers have, and the amount of molecule passed through both these also varies. Thousands to millions of ions can pass through when a channel opens, while one or small amount of molecule only pass through carrier molecule. Channels are bigger class of transporter, which is further subtyped into two: voltage-gated ion channel and ligand-gated ion channel. Voltage-gated channels are most commonly seen in neurons and in many other types of cells. They are constituted of several subunits arranged in a way so as to form a central core which allow ion to pass through them. A change in electrical potential near the channel activates it. For example, this occurs in neuron where rapid and coordinated depolarization occurs in response to triggering voltage alterations. These directionally propagate electrical signals through axon and at synapse, thus messages are transported in brain. These channels are ion specific, although similarly sized, and charged ion pass through at a time. As you can see in the illustration, the sodium and potassium channel in nerves and muscles are voltage-gated channels. Calcium channels at the presynaptic nerve ending are also example for voltage-gated channels. This is another type of ion channel protein which allow for the passage of anion and cation such as sodium, potassium, calcium or chloride ion through cell membrane. The major difference with the former is the stimuli, and in this case the movement of ion occur in response to binding of a ligand or a chemical messenger such as neurotransmitter or ATP. These channel proteins have two domains, an extracellular domain and a transmembrane domain. The extracellular domain has a ligand binding site location for allosteric binding, while the transmembrane one create a pore region within the spanning region of the membrane. Modulation of ligand-gated ion channel can also take place through channel blockers, ion or membrane potential. Ligand-gated channels are classified into three superfamilies, namely cysteine loop receptors, ionotropic glutamate receptors and ATP-gated channels. Let's discuss them briefly. These receptors are named after a characteristic loop which is formed by disulfide bond between two cysteine residues present at the N-terminal end of the extracellular domain. They can be subdivided on the basis of the type of cationic or anionic ion which they conduct, and also into families as defined by the endogenous ligand. Usually, these receptors are pentameric in nature, which means there are 5 subunits, which each subunit containing 4 transmembrane helices. These helices constitute the transmembrane domain and a beta sheet sandwich type, extracellular, N-terminal and ligand binding domain. The figure showing nicotinic acetylcholine receptor is a type of cysteine loop receptor. These are the receptors which bind to glutamate, a neurotransmitter. I hope you all know what a neurotransmitter is. These are chemical messengers that transmit impulses across the neuron. This channel forms tetramer, wherein each of the subunit consists of an extracellular amino-terminal domain, an extracellular ligand binding domain and a transmembrane domain. Binding of glutamate to the tetramer assembly activate this ion channel. AMPA receptors come under this superfamily. The third subclass of channels are ATP-gated channel. As the name suggests, this channel open only upon binding of ATP, adenosine triphosphate. Structurally, they form trimers. 2 transmembrane helices are present per subunit. Both C and N-terminal lie on the intracellular side. There is also an extracellular domain. As you can see, P2X receptor is a type of ATP-gated channel. We discussed the subclasses of channel till now. Let's see what are the different type of carriers present in the cell. These are integral membrane protein that mediate the movement of substances by facilitated diffusion or active transport. The carriers present an import or export site for the molecule to be transported and may also undergo a change in conformation which is induced largely by the substrate. These movements are hence called carrier-mediated transport. Only one substance or one group can be recognized by each carrier protein, and carrier-mediated transport is further divided into 2 types, passive transport and active transport. The word passive means it does not require an input of cellular energy. Transport occurs across an electrochemical gradient. Example include transport of sugar, amino acid, nucleoside, purine and pyrimidine, and more sodium calcium exchanges, chloride bicarbonate exchanges and many more. The rate of passive transport depends on the permeability of the cell membrane, which in turn depends on the organization and characteristic of the membrane lipid and protein. Passive transport include 3 types of transporters: Uniporter, Antiporter and Symporter. Uni means 1. So, if a carrier protein binds to only one substrate molecule at a time, which mediates its transport, such transporter is a uniporter. The transport is along a concentration gradient and hence there is no need for energy, and thus it is a passive transport. Example, the transport of glucose, amino acid, nucleoside, monocarboxylate transporters, and so on. This is another type of passive transporter. It is a cotransporter, which assists the transport of two or more different molecules or ion in opposite direction across cell membrane. Therefore, they are also known as exchangers or counter transporters. This is also called a cotransporter and an integral membrane protein, but this is associated with the movement of two or more different molecules or ion not in opposite direction like an antiporter, but here in the same direction across a plasma membrane. So students, so far we discussed on passive transport. Now let's discuss on the second type of carrier transport which requires energy, and that is active transport. Unlike passive transport, active transport involves the movement of molecules across the cell membrane against concentration gradient, which requires energy. Active transport is further subdivided into two types depending on the type of energy used, that is primary active transport and secondary active transport. You might be curious to know what is the major difference between primary and secondary active transport. Primary active transport is also known as direct active transport since it uses metabolic energy, that is ATP, directly for transporting molecules across the membrane. So what are the examples? P-type ATPases, sodium potassium pump, etc. Secondary active transport is otherwise known as coupled transport or co-transport. It does not involve direct ATP coupling. Pumping ion in and out of the cell lead to the formation of an electrochemical potential difference, and this electrochemical gradient is used to transport molecule across a membrane. Example of such transporters are cotransporters of sodium potassium, neurotransmitters and cations, sugar and cations, amino acids and cations, cation ATPases, sodium, potassium, calcium, hydrogen, and multi-drug resistant transporters like phosphoglycoproteins. So till now we discussed various types of transport of micromolecules. Now let's discuss on protein mediated transport of macromolecules. Transport of macromolecules such as protein, polysaccharides, polynucleotide, etc. across the membrane occur mainly by two independent mechanisms. They are endocytosis and exocytosis. As you all know, endo means movement towards the cell and exo means movement outside the cell. Endocytosis. It refers to the intake of macromolecules by the cells. It involves active transport wherein a cell engulfs macromolecules in an energy using process. Endocytosis pathway can be of four types: Cladrine mediated endocytosis, Caviola, Macropinocytosis and Phagocytosis. Let's discuss on these. It is a process mediated by small vesicles of approximate diameter 100 nanometer. These vesicles are known as Cladrine coated vesicles or CCVs. These vesicles are coated by a protein complex associated with cladrine, which is a cytosolic protein. These are found in all cells and form pits or domain in the plasma membrane called cladrine coated pits. Large extracellular molecules which have different kind of receptors that can aid the process of endocytosis of ligand can be concentrated in these cladrine coated pits and then enter into cells. These are small flask shaped pits about 50 nanometer in diameter, present in the membrane on many cell type surfaces. These are non-cladrine coated and consist of another type of cholesterol binding protein called caveolin. They are abundantly found in smooth muscles, type 1 pneumocytes, fibroblasts, adipocytes and endothelial cells. receptors in caviolae are also said to be involved in mediating the uptake of extracellular molecules in bulk. Pinocytosis, or well known as cell drinking. This usually occurs from highly ruffled plasma membrane region. It involves the invagination of the cell membrane to form a pocket, which later detach off into the cell forming a vesicle. The diameter of the vesicle is about 0.5 to 5 micrometer and is filled with a large volume of extracellular fluid and molecules. And these vesicles move into the cytoplasm where it may fuse with other vesicles, namely endosomes and lysosomes. This is a process which involves binding of a cell to a particulate matter like dust particle, cell debris, microorganism and even apoptotic cells, followed by its internalization. Phagocytosis or bulk cell eating occur in case of whole organelles like ribophagy for ribosomes, mitophagy for mitochondria, pexophagy for peroxisomes and xenophagy in case of microbes. These are the various types of endocytosis. Now, exocytosis, which involves the release of macromolecules from cell to the outside. It is an active transport process. During exocytosis, the vesicular content, as you can see in the figure, are transported by secretory vesicles across the cell membrane into the extracellular space. These contents within the membrane-bound vesicles may include soluble proteins and membrane proteins or lipids, which are to be secreted into the extracellular environment. Exocytosis is also of three different types. They are calcium-triggered non-constitutive type, which is also known as regulated exocytosis, non-calcium triggered constitutive type and vesicular exocytosis. This refers to the process of exocytosis that occurs in neuronal chemical synapse. It is triggered by calcium and mainly serve as interneuronal signaling, that means between neurons. This type of exocytosis requires an external signal, a sorting signal on the vesicle, a clathrin coat, as well as an increase in intracellular calcium. That's why it is known as regulated exocytosis. This type is also known as constitutive exocytosis. This type of exocytosis serves to release the components of the extracellular matrix or delivery of newly synthesized membrane for incorporation into the plasma membrane after fusion of the transport vesicle. This third type of exocytosis has been very recently observed in gram-negative microbes. The plasma membrane in these microbes is pinched off as vesicles called bacterial outer membrane vesicles or OMVs and are used for translocating biochemical signals into host cells, eukaryotic or other microbes. Though these are transferred as bacterial outer membrane vesicles, this mode of exocytosis is known as vesicular exocytosis. Dear students, in this module, we have discussed the role of protein in mediated transport. Transporter proteins are involved both in micromolecular and macromolecular transport, among which we discussed channels and carriers are the major micromolecular transporters, while endo and exocytosis as the major modes of transporting macromolecules. Each subclass and its characteristics have been discussed in detail. To conclude, in simpler terms, we can say that the need of a cell for various metabolites are sensed. These transporters then play a role in internalizing the required molecules or extruding the unwanted, like in case of toxins, etc., to maintain the balance through these transport mechanisms.